Lumit immunoassays are bioluminescent assays that can detect any protein and are easier and faster than conventional ELISA tests. ELISA tests involve multiple incubation and wash steps. Often taking hours to get results. Lumit immunoassays eliminate the need for all washes, reducing the procedure to a few simple steps. First, chemically label selected antibody with large bit or small bit. Then, mix labeled antibodies with sample. This brings large bit and small bit subunits together to form a functional luciferase. Add Lumit detection reagent to produce luminescent signal proportional to analyte levels. Measure signal using a plate reading luminometer, such as the Glomax Discover. Get results quickly at the push of a button using a preloaded protocol. The whole process, from sample to results, takes 1 hour or less. Use Lumit immunoassays for fast, simple, and sensitive analyte detection.
The Caspase Glow 1 inflammasome assay is a fast, simple, specific assay for detection of active caspase-1 as a biomarker of inflammasome activity. The protocol is easy and straightforward, involving only 3 steps: add, mix, and measure. This is in contrast to the labor and time-intensive protocols associated with traditional Western blots and ELISAs for detecting caspase-1 or IL-1 beta. Those assays also lack selectivity for active forms of the enzymes, unlike the Caspase Glow 1 assay, which is selective for active caspase-1. The inflammasome is a protein complex that is induced by diverse inflammatory stimuli. Innate immune cells respond to pathogens and other danger signals by forming the inflammasome and converting pro-caspase-1 zymogen into catalytically active caspase-1. In this assay, active caspase-1 cleaves the selective ZWEHD substrate and releases luciferin. Luciferin then reacts chemically with luciferase, resulting in light production. The amount of light produced correlates to caspase-1 activity. Inflammasome research has never been this easy. Experience greater productivity with the quantitative, selective, and time-saving Caspase Glow 1 inflammasome assay. Learn more today.
When assessing dose response, multiple plates of cells, media, reagents, and expensive treatments can be reduced to a single plate using the real-time Glow MT cell viability assay. Viability assay development using an endpoint method requires multiple plates. Assay development using a real-time method uses only 1 plate and much less reagent to measure cell viability continuously out to 72 hours using a simple plate reading luminometer. It's as easy as adding the reagent at any point during the assay and measuring luminescence as many times as desired for the same well. The MT cell viability substrate enters the cell, and only metabolically active cells can reduce substrate, which exits the cell and becomes a substrate for luciferase. Dead cells cannot reduce the substrate, therefore there is no luminescence. With real-time Glow, there is no guessing at optimal time points to capture response. Data is collected in real time, so you never miss out. Since the cells are not lysed, you can respond to what you see in real time by using the same sample to measure cell death with an assay like CellTox Green, or measure apoptosis, stress response markers, or even analyze RNA. The real-time Glow MT cell viability assay allows you to collect more relevant data per sample with less time and effort using fewer plates of cells.
Lipid metabolism research often requires measuring lipolysis in cells. This can be done by detecting the amount of glycerol in cell media. Traditional methods to measure lipid metabolism in cells often requires organic extraction, which is time-consuming, tedious, and involves toxic reagents. Our lipid metabolism assays use sensitive bioluminescent technology and novel detergent reagents to allow detection of glycerol, triglyceride, cholesterol, and cholesterol esters without organic extraction. The protocol is simple and fast. Pipette a small amount of media from cell samples into a multi-well plate, then add glycerol lysis solution, or add lysis solution directly into sample wells and shake. Add glycerol detection solution to each well and shake. Incubate for 1 hour at room temperature. Record luminescence of samples using a plate-reading luminometer such as the Glomax Discover. Pre-programmed protocols on the Glomax Discover make data collection super easy, fast, and accurate. Glycerol concentration is calculated by comparing luminescent signal to a standard. Glycerol levels correlate with lipolysis in cells. Measuring lipid metabolism is simple and fast using our bioluminescent assays, which use add and read protocols and are compatible with 96 or 384 well plates. When immunogenic cell death occurs, cells release ATP into extracellular space. Measuring the amount of extracellular ATP in real time can tell you when and how immunogenic cell death is induced after treatment. The Real-Time Glow Extracellular ATP assay measures extracellular ATP based on bioluminescent detection using a simple protocol. Plate and treat cells. Add assay reagent to cells. Measure the same plate at each time point for 24 hours or more using a plate-reading luminometer such as the Glomax instrument. By recording luminescence, you can easily determine dose-dependent timing of ATP release. The Real-Time Glow Extracellular ATP assay allows simple, real-time monitoring of immunogenic cell death or other signaling pathways and is compatible with 384 well plates.